So if you think about this whole course, what are the major biological problems we try to discuss? The first thing is transcription regulation and transcription factors. We started the course with gene expression, right? Just RNA expression, but RNA is a descriptive measure. You just know, whether the gene is up or down. The question is why they are up and down. And so one major question that everybody care about, starting from embryonic stem cell differentiation into anything, neuroprogenitor cells, into or blood differentiation into different white blood or red blood cells, and also cancer development is all disease or is all related to transcription regulation, and transcription factors are very, very important in this. Of course, once we care about transcription regulation, you see that the same transcription factor can have very different effect in different cells. Then epigenetics comes around. We understand that transcription factors and epigenetic profile is like a chicken egg problem. They each have big influence on the other. So the collection of all the transcription factors establish the epigenetic status, but all the epigenetic status of the cell determine how much a transcription, or influence how much a transcription factor can bind to some locations but not other locations. And in the process, the transcription factors also work together with the epigenetic factors, the histomark or DNA methylation readers, writers, and erasers, right? They add the mark, erase the mark, or they read the mark, or there are also chromatin remodelers that move nucleosomes around. These are all definitely important factors in maintaining the system balance. And then we talk about human genetics and disease susceptibility. In the early days, it's just you do a lot of SNP arrays or whole exome sequencing or whole genome sequencing, and then you do a gazillion chi-square tests, kind of the simple thing. But later on, you realize there are also good statistical problems such as population stratification. How do you get more sensitive results? But right now, the whole GWAS or rare disease field are looking into using gene regulation or epigenetics to help them understand why do these SNPs create the phenotype or the disease. And so the whole GWAS world, especially the pioneers in the field, are working closely with scientists working on gene regulation to make sense out of their GWAS hits. And then for cancer genomics is also all the mutations, right? Initially, you can get a paper published by sequencing a few hundred tumors, get the recurrent mutations, but now everybody also cares about why does this mutation cause the cancer? And if this does cause the cancer, then what? What are the drugs? What are, whether the mutations make the tumor more sensitive to some other genes and things like that, right? So those are the important biological problems. In terms of the genomic techniques that we discuss in this course, RNA-seq and single-cell RNA-seq, right? For gene regulation, for motif analysis, we very briefly discussed PBM and CELEX, which is a way for us to get a collection of different transcription factors motifs. And also ChIP-seq is where you can map the transcription factor in a particular cell or tissue. For epigenetics, we talk about bisulfite sequencing. You can also use ChIP-seq for histone modification. We talk about DNA-seq or ATAC-seq. This is a way to look at all the open chromatin, which is a collection of all the transcription factor binding sites. And then we talk about single-cell ATAC-seq and also Hi-C. There are people also working on better ways such as cut and tag or single-cell ChIP-seq, and maybe by next year we'll have single-cell ChIP-seq data coming, or single-cell cut and tag. So for GWAS, as we mentioned in the early days, people used SNP arrays, but also now people are doing sequencing from whole exome or genome-wide. And for those studies, you can either do the case control study or the family-based association studies. And then, for genome sequencing, you can look at the germline sequence, which is DNA or those mouse swabs, just get some epithelial cells you can send to companies to do your germline sequencing. Germline sequencing is mostly to look at the disease susceptibility. Somatic mutation is, when the patient has a biopsy and pathology has already diagnosed this patient as having cancer, and you compare the tumor biopsy DNA versus the blood DNA to really look at the mutations. And based on that, you can match the patient with specific drugs. And in order to really match the drugs well for like precision oncology, you can do either genetic screens or chemical screens to see, this mutation is sensitive to that drug or inhibition of another gene. And then I think we are seeing more and more of this combination of different genomic techniques. For example, CRISPR screens can be added to single-cell RNA-seq. So you can knock out different genes, and they might have a different gene expression, but then you can do this in a pool and do single-cell RNA-seq, right? We, in this course, also talk about people trying to, from the same tumor tissue, do single-cell RNA-seq and single-cell ATAC-seq in different cell, the same tissue, different cells, but currently there are techniques being developed to do single-cell RNA-seq and single-cell ATAC-seq on the exact same cell. They share the same cell barcode, but the RNA is used for RNA-seq and the DNA is used for ATAC-seq. So later on you can match these really one-to-one together. There are also proteomics, combined with single-cell RNA-seq, so that you can use protein markers on the cell surface to select the cells to do single-cell RNA-seq. And also, now people are using imaging, trying to have spatial transcriptomics. It is possible, but by next year, we will see more spatial transcriptomics data. So the idea there is, if you have a chip, and on the chip you have different barcode, each barcode you know the exact location. And if you have a tissue which has RNA, you just put them together, and then the RNA will be attached to the barcode on this chip. And so you can make the sequencing library. And so each of the RNA not only will be amplified, it will also carry the barcode coming from here in a particular location. And then at the end, you put all the RNA together for sequencing. But based on the barcode, you will know where it is on the original, on the original chip. Then you will know, okay, this cell is next to that cell, or what are the neighborhood information, which is really cool. And I think if this technology is getting good enough, robust readouts, then you do not need droplet anymore, right? So, but right now, these are all technologies under very fast development. And then in this course, we also hope to teach people statistics and computer science algorithms. I hope that you have a good exposure of R, bioconductor, and Python programming. For statistics, we talk about normalization. How do you make sure you have a normal distribution? You can test this by QQ-plot. We mentioned many different statistical tests, t-test, a chi-square test, Karmanov-Smirnov test, Mann-Whitney-U test, and so on. One important thing to know is with genomics, because we are testing every gene or many genomic intervals or many SNPs, we need to do multiple hypothesis testing correction. And for that, you can do Bonferroni correction, which is very conservative, or you can look at false discovery rate, which is less conservative, will probably give you a reasonable number of good hits. And then we talk about hierarchical models or generalized linear models. How do you borrow information from many other genes to give you a trend? How do you, for example, when we do differential RNA expression, if you don't have enough samples, you would not estimate the variance very well. How do you use the variance estimated from all the genes together to give you a trend? And based on that, prior updates your estimation for the variance for each gene, right? How to do this well. For clustering, we taught you guys how to do k-means clustering, hierarchical clustering, principal component analysis, how to do UMAP is kind of also clustering, but also projection approach. You can visualize it together. And also Surat have this graph-based community modularity network approach for clustering. And then for machine learning, we talk about support vector machine, we talk about random forest, neural network, and how to use a regression-based approach for statistical machine learning, and how to use lasso to select features, right, to go through the logistic regression. And then in the read alignment, we talk about Boris-Waller alignment. And also in motif analysis, we talk about expectation and Gibbs sampling. In segmenting the genome, we talk about hidden Markov models and how to use dynamic programming. And also in subtype analysis, we talk about survival analysis. This is more statistics, right? So, I want to mention that in general, in bioinformatics, you see this trend. When a new technology is developed, usually, initially, CS people are faster. They can code fast, they can develop pipelines, they can do the pre-processing, and also anything that is just a read alignment, you know, you cause some mutations, or those usually the CS scientists do better. But later on, when these are done, you're dealing with many samples. You're trying to make a comparison, differential level, how to do normalization so that the differential analysis is more correct, how to adjust for batch effect. You know, those, anytime you deal with numbers on many samples, the statisticians will have an edge. They know how to do these estimations well, model the data distribution, and then give you more robust hits and remove the false positives, or alleviate the multiple hypothesis testing problem. And so then after the statistical methods are developed, then a lot of people develop are adopting the technology to do the assays. Then there are also people collecting data, create databases, and try to utilize public data to do hypothesis generation and make sense out of this data. And you can see that it's the people who understand the biology. They know how to ask questions from all the public data. They can do better. And so even though all of them can be computational biologists, they might be playing a different role in this process, which is very exciting. So everybody has a chance to do well in computational biology. We also talk about some bioinformatics algorithms in the course, right? For geontology and pathway analysis, we talk about David, gene set enrichment, and also single sample gene set enrichment. We talk about batch effect removal using Combat for sequencing alignment. You need to use FastQC to QC your raw data. You can use BWA to map the reads, or STAR to do the RNA-seq mapping, or you can use Callisto or Salmon for the pseudo alignment. You can quality control your RNA-seq with RCQC. Yeah, so Salmon and Callisto. We can look at the DE-seq to do differential expression analysis. You can also, by the way, use Lima for batch effect removal. For single-cell RNA-seq analysis, we talk about Surat, which have a lot of features. For ChIP-seq or DNAs, we talk about Max, and also Systrom is a good database. And the single-cell attack-seq, Surat has some solutions. Many groups are developing tools. Our own group has a new tool called Maestro that we would encourage folks to try it out. And for mutation sequencing, we talk about Mutect, CRISPR screens. Yeah, you will be using MAGIC in homework 6. And for immunology, PolySolver is for HLA typing. NetMHC is to predict which of those mutations will be presented on the cell surface by the HLA of the patient. Timer for immune cell infiltration estimate. Trust for TCR, BCR extraction from the bulk sample. And the TIDE for estimating these different immune cell types for immunotherapy prediction. And by the way, there is no way we would have taught you every possible algorithm. We try to give you a general idea, with this type of problems, you should probably do things roughly this way. But these algorithms are being developed all the time, especially the newest technologies, such as single-cell RNA-seq or single-cell ATAC-seq. There are more and more methods developed, and we can't possibly teach you everything. But then hopefully from this course, you won't be afraid to learn a new algorithm. You just pick it up, read the README, test it out, right? Troubleshoot. And hopefully this course would have given you that experience. What we don't want to give you is line by line, command line to say, okay, you just do this, then you only know how to get there. So bioinformatics is a lot of trial and error. We hope that we teach you the experience that some homework you can see is easier. Otherwise, maybe it's a little harder. You have to troubleshoot. That's very, very common for bioinformatics. And so we hope that this course will give you the foundation so that you will never be afraid of data again. All you need to know is, okay, let's look at this problem. What are the tools I have to master? And if there is no tool, well, maybe you can develop one later, right? In the very early the first lecture of the course, we mentioned the different level of bioinformatics. I want to kind of go over this again. I hope you, by this time, you have gone through the course, you will understand these levels better. Level 0 is modeling for modeling's sake. Some people want to look at bioinformatics problems, and yet they are not providing a solution that anybody cares. It is not helpful for computational biologists. It's not helpful for biologists, right? They are usually not published in high-profile journals and not very widely read or cited. We don't want you guys to do level 0 bioinformatics. It's a waste of resource. Level 1 bioinformatics is roughly where we started in this course, right? How do you analyze some data from existing tools? You can write simple scripts to just transform the data in the right format, but most of it is using existing tools. It could be that you are the student in an experimental lab, you've taken this course, or maybe you have generated your own data, right? How do you analyze the data and make sense out of this? And you have to ask yourself, or if you are a computational lab, you might be collaborating with a biologist. And in order to decide whether somebody is doing a good level 1 bioinformatics, you have to ask, do they use this to solve a very important biological problem? And are there existing algorithms to analyze the data? How much programming does this person need to do in order to get there, right? Maybe it's all just using tools. And does the analysis and experiments inform each other? I think a good level 1 bioinformatics is the data generator initially in the study already talked to the computational biologist to say, I'm interested in doing this experiment. Do you think it's a good design? Computational biologists say, okay, maybe you should, add this experiment or not do the experiment this way. And then after the data comes, you analyze the data and say, based on this, I'm seeing some hits. And then the experimental group would go and validate this. And then they generate new data, which then the computational computational biology analyze it again and generate new hypotheses. So you can see the experimental, computational, experiment, computational, experiment, computational, alternating in papers. That means it's a real hypothesis-driven, really collaborative efforts to identify a biological phenomenon. There are cases with level 1 bioinformatics. All the data are generated. The experimental biologists already had the hypothesis. They are very clear where they want to go. They just say, 1, 2, 3. You generated the figures, you analyze the data, generate the figures, and support my hypothesis. Now, those are not so great collaborations, actually. So you want this to really feed each other. And also you want to see, does the study have real biological findings? We don't want a lot of studies to have just a lot of data, not really new findings, and just kind of the informatics is to make sure that data is at least pre-processed and past QC, that's it, there it is. It's a big data paper. There are a lot of data papers published, by the way. Does the experimental group value the bioinformatics contribution? Did they actually use informatics to make a new finding, a significant finding? And also, sometimes an experimental group might have a hypothesis, which then the computational analysis says, no, this hypothesis is wrong. Is the computational group willing to revise their hypothesis and follow the data rather than insisting on their original hypothesis, right? And in order for bioinformaticians to do this well, of course, you need to be familiar with all the different bioinformatics algorithms, but also understand the biological problems and be willing to use data from orthogonal approaches to validate a hypothesis, to help refine a hypothesis so that the experimenter can then hone in on the right hypothesis, then do the right experimental validation. They will get to the result faster. And especially, I think this is especially important if the data is pointing to an alternative hypothesis, which might be unexpected from the original experimenter. You need to understand the biology and use other data or previously published papers to substantiate the new hypothesis to convince your collaborator that the data is pointing to a different direction. Yeah, and so you can really need to learn the biological domain knowledge, okay? And motivate method development. Yeah, so we a lot of times collaborate with experimental biologists to kind of help us to learn biology. Like, say, initially I wanted to do immunology. I don't know much about immunology, but I know computational analysis, so I can collaborate with an immunologist. I can contribute my computational expertise, and they can kind of teach me immunology. That will be kind of a win-win collaboration. So the danger here is try to avoid being a data janitor. This could be a danger if you are the lone computational biologist, analyst within a big experimental group. All the data in the lab are coming to this one person who is just too busy, even just to QC all the samples. Basically, the experimental lab members are lining up to have this one guy helping them with processing all the data. Then you just become a data janitor. You have no time to really think, to really learn as much, and also really contribute in a meaningful way. You won't really be involved in hypothesis generation, and also kind of really look at the results, say, what does this really tell us? I think this is important for a bioinformatician, not only to make sure that the data pass QC, but say, what does the data tell us? Does this make sense? Does this change our understanding of the biological problem, right? That's really important. So even for level 1 bioinformatics, we are teaching you all the basics, but we ourselves are still engaged in similar type of research. But from this, we contribute a lot to the experimental group, but we also learn a lot ourselves, which is, I think, a very satisfying experience. Level 2 bioinformatics is, I think that's what makes computational biologists a computational biologist or a bioinformatician. Usually, if you're a level 1 bioinformatician, you only benefit your own group or your collaborator. But if you are a level 2 bioinformatician, you develop algorithms like DESeq2, the BWA, you develop Max, right? So you can help many, many users in the community. You can develop a database like DepMap or CBio portal, right? So those millions of people use those ones, like hundreds of thousands of scientists might be using those tools. That's really powerful, right? How to develop algorithms or databases for mining biological data. And so usually these scientists need to have a keen eye on what are the new techniques out there. So if there's a new technology developed, very often the technology developer is an experimental group. They don't have the capability to develop algorithms, right? But these type of new algorithms are developed all over the world. For a computational biologist, you have to decide your resources are limited. If I develop tool for X-seq, I will not have time to develop tool for Y-seq, right? Which one do I go for? you have to know which one will really have an impact and can answer more interesting biological questions. And you have a unique approach to develop a tool rather than just doing it quick and dirty. So those are some of the considerations. You need to make sure you use sound statistics. Also, the tool needs to be very, very fast or user-friendly so that people will use it. And a lot of times as the data, like for example, for RNA-seq in the early days, people sequence 3 million reads, then they sequence 10 million and 20 million. And now they easily sequence 50 million reads. And so the data challenges will change over time. So the tool developers will continue to have to maintain and update the algorithm with new data and new challenges in order for their tool to continue to help the community. And so in the early days, you might be able to provide one small solution, just one thing. But nowadays, a lot of times when you collaborate with a biologist or say tools like Surat, it's not just one solution. It is a complete solution, right? The developers, they understand biological problem. They incorporate, they develop a lot of the tools already, but they also incorporated other solutions developed by the statistical world or computer science world or the machine learning world to really incorporate all in Surat or ScanPy or these tools so that people can just go there and just call out the different functions to really solve all their problems. And that's really, really important. And so computational solutions that only solve one problem is like small aspect is useful, but in terms of impact might be less, it might be limited, but also their tool might get eaten up by those big, complete solutions and be shadowed. And a lot of these computational solutions may or may not need to publish in high-profile journals. From our experience, whether the paper is published in Genome Biology or Nature Biotech or NAR probably doesn't matter as much, even though their impact factors are quite different. The key is the algorithm really widely used and cited? Do people use it to make real findings? Do you provide the benefits to the whole community? That's really important. And in order to do that, all of these above factors matter. Do the tool developers understand biology, provide the right features to answer the questions? Does the tool use sound statistics, efficient coding and user-friendly features, right? All of this, and do they continue to keep updating the tools? These are very, very important. And very often they don't even get a new paper from the update of those tools, but they still do it. And that's why over time they do get acknowledged by the field for their long-term commitment to those tools. And so that's what we call doing well by doing, or do well by doing good. You do good to the community, eventually the community recognizes the contribution of these long-term commitments. And there are also level 3 bioinformatics, which is how do you make biological discoveries from public data integration and modeling? This is slightly different from level 1 bioinformatics. Usually level 1 is your own lab or your collaborator generated some data. They already might have some hypothesis. You just help them analyze the data to get this paper published. So you start from unpublished data. Whereas level 3, you start from all the public data. If you're like, I'm interested in knowing what will influence T-cell exhaustion. Can we identify a new drug that can be combined with previous drug to treat some disease, or some basic science questions? And because there's just more and more biological data available in the public, people can now really understand these problems much better and use data. When I was a PhD student, we have to beg our collaborators to say, can we collaborate so I can look at your data? We want to be a level 1 bioinformatician. But nowadays, things are changing. There's so much data in the public. We can just integrate public data if we understand enough biology and we understand the data. we can integrate public data and make discoveries ourselves. And this is real data mining. Computational biologists ends as a biologist. And now we can be a true biologist by making biological discoveries ourselves. You need to have some unique biological angles. You can use your algorithms that's unique, but it could be your own algorithm. It could also be other people's established algorithm, but you use it in a unique way to answer some interesting biological questions. Yeah, so make sure that you are using actually this to do real discovery, not just putting numbers on theories. So there are sometimes existing theories, and there are people just say, oh, look at all the public data. It really agree with the theory again. That's not very interesting. A lot of these, sometimes because we are integrating public data to make discoveries, reviewers do want to see experimental validation. And those experimental validations can be done directly by the informatics groups. Nowadays, it's not uncommon for a computational biology group to have a small component of experimental biology, experimentalists in the lab. But also you can work with your experimental collaborators. And that's why we engage in level 1 bioinformatics work. You know, we help our collaborators when they have a hypothesis, but when we have a hypothesis, they might also help us validate those experimentally. In recent years, we also see more and more level X bioinformatics, which is integrative papers from big science consortium. These are, you know, national or international consortium. They generated a lot of data, and then 3 years is done, the grant has ended, then the whole consortium published usually 10, 20 papers in one batch in a series. These are pretty, you know, I think these data are very valuable. Writing those consortium papers are very exhausting and time consuming. And usually there's discussions with high profile journals ahead of time to say, okay, Nature will say, well, take these 5 papers. And if we don't want those papers, the remaining one can go to another journal that they talk with together. And all the papers are coordinated. But you know, some of these times the contributions are sometimes tricky. I mean, the people who got good authorships definitely worked harder towards that. But sometimes whether the finding is, you know, the major finding was coming from that is questionable. So trainees can be involved in this, but just be careful that you need to learn how to stand on your own as a computational biologist, because supposedly if a trainee was in a consortium initially, they can get first author Nature Science papers, but one day they have to go out, and if they are no longer in that consortium, they may be cut off from such data. How do they stand on their own when you don't have data feeding to you? Can you come up with ideas to publish your own methods or publish your own discoveries, right? So I think a good bioinformatician should do level 1, 2, 3, and X, all of them. As a student, I would say when you are a PhD student, you know, try to do more 1 and 2, but when you are a postdoc, you know, try to do more 2 and 3. Level X sometimes depends on which lab you are in. And also people with more CS and statistics background should do more level 2, but if you have a biology background, you can do level 1 and directly to discovery in level 3, right? So it's a different balance. Okay, so that's that.